Hello, welcome to this new lecture series on explicit memory storage. I'm Andersen, and this is the very first lecture in that lecture series of, on explicit memory. So let's get into that. Let's start our lecture. By the way, this is a very beautiful painting by Dr. David Swat. It shows, a layer of the hippocampus called stratum pyramidal. I'm not sure if I'm pronouncing it correctly, but it's a layer of pyramidal cells in the hippocampal cortex. So let's talk about the explicit memory storage. I've divided this lecture series into 3 sections because there's a lot to talk about actually. Well, section 1, which is this section, I'm going to talk about hippocampus, its macroscopic anatomy, its gross anatomy, and also its a little bit about its synaptic circuits. Then I'm going to talk about the induction and expression of LTP in different pathways in the hippocampus. And finally, I'm going to introduce a concept called silent synapses in the first section. In the second section, I'm going to focus on early and late LTP and the role in memory formation and consolidation. And finally, in the last section, in the final section, I'm going to introduce brain microcircuits, the synaptic organization of the brain, and I'm going to talk about the hippocampal microcircuits. Okay, so we had this chart in previous lectures, in the introductory lecture, in the very first introduction lecture, in the very first section on, first section of implicit memory lectures. And so here we have it again, but, these are, subtypes and different types of long-term memory. I already talked about the implicit or non-declarative memory. And now, in this lecture series, I'm going to talk about explicit or declarative memories. Semantic memories about facts, for example, definition of a word or the name of places and people. And then episodic memories, memories about our experiences and events, different events. So this is classification. I'm not going to focus a lot on that. Okay. Well, I mentioned that a lot in previous lectures, that the main focus of all of these lectures is just on the cellular and molecular aspects of memory and memory formation or storage. Obviously I have to talk about just the synaptic connectivity of the hippocampus, but the gross or the macroscopic anatomy of the hippocampus and hippocampal formation in general is so magnificent that I couldn't convince myself not to talk about it at the beginning of this lecture. So I devoted almost half of this lecture to just the macroscopic and general anatomy of the hippocampus and its associated structures in the brain. So now I'm going to just show you, a general view of the hippocampus and where it is located. This is the organ we are all obsessed with, the human's brain, and there are some structures. We have, we have a transversal cot, and you can see that we have the corpus callosum here, one of the commissural fibers of the brain connecting to cerebral hemispheres. We have, exactly above the cerebral corpus callosum, we have the cingulate gyrus. Here we have the head of the caudate nucleus. A caudate nucleus is part of the basal ganglia in the brain. I wanted to present a new lecture series on basal ganglia, but I decided not to because, this is enough. I may do that in the future. But anyway, so here you can see the thalamus, which is of course cut in half, and beneath its anterior or frontal part we have hypothalamus, And then we have these mammillary bodies. You're not shown clearly here. I'm going to actually show a better picture later of mammillary bodies. But then we have mammillary bodies of the hypothalamus, and then we have this structure which originates from the mammillary bodies, and then we have it wraps around the thalamus, and this structure is called fornix. So we have the fornix. All of these. I'm just reviewing neuroanatomy for myself. Many of you, Professor Yule Jackson McMahon, Professor Gilbert, Professor Eric Kendall, all of you've studied anatomy, MD, and of course Professor Matthew and Ashmore. All of you know neuroanatomy much better than I do. So I'm just reviewing this for myself. And here we have the fimbria, and fimbria, it's a shared structure between hippocampus and the fornix. And finally we have this round some sort of a seahorse structure here. By the way, this is the temporal lobe, okay? And in the middle of that temporal lobe we have this structure, and I've highlighted that, and this is the hippocampus. So I just wanted to show you a very, very general position. This is a real picture of hippocampus in a real brain, a real human's brain. And now let's talk about the structure of the hippocampal formation and the hippocampus itself in much further detail. But before I do that, I have a brain model here. It's a very good brain model. My father bought that for me for my 17th birthday, I guess, and it was a great gift. And so we have, you can see that I have the left cerebral hemisphere here, the corpus callosum, the cingulate gyrus, the thalamus and hypothalamus, and let me just point it with this. This is the mammillary body. I don't know if you can see that very well or not. This is the mammillary body. And this is the temporal lobe. And I told you that the hippocampus is located in the temporal lobe. And let's take a deeper look at the temporal lobe. This is the temporal lobe. And you can see a part of the fornix here. A part of the fornix. Let me just show it in front of the camera. I guess here, here you can see that better. Yes, you can see that much better here. So we have the part of the fornix here, and then we have the fimbria. This is the leg or crus of the fornix, and then we have the fimbria of the fornix. Here we have the parahippocampal gyrus, okay? And you can see the hippocampus here. This is the hippocampus. Okay, let's get back to our video. I'm very glad that my model has hippocampus. Okay. So that was the hippocampus in here. You can see that it is located in some sort of a cavity, and this is ventricular cavity. In your brain, we have some cavities called ventricles, and they contain cerebral spinal fluid, or a CSF, and that fluid protects the brain, and it has some nutritional value, I guess, to the brain. One of the main functions it does is to protect the brain from shocks. Okay, so physical accidents and physical shocks. So this is the relative position of the hippocampus in my model. I just wanted to show you a 3D version of that. Okay, now let's talk more about it. So this is another view. Many of these drawings are from the book called The Atlas of Human Anatomy. Yes, Atlas of Human Anatomy, by Professor Frank Netter, a wonderful anatomist and physician. So let's get to this. Okay, So we have removed, not we, but, the anatomist, the neuroanatomist, have removed the corpus callosum, but they have left a part of that. This is the genu of the corpus callosum, the anterior bend, okay, the bend in the frontal part of the corpus callosum. This part. This is the genu. This is the body of the corpus callosum. This is the splenium, the posterior bend. Can you? Yeah, you can see that. Okay. Anyway, so this is the genu of the corpus callosum, and this part is the posterior bend or the splenium of the corpus callosum. And you can see, again, we have the head of the caudate nucleus. Then we have the column of the fornix that ends to the mammillary bodies of the hypothalamus. And then we have the body of the fornix, the crus, or the leg of the fornix. The plural of that is crura, or the legs of the fornix. And then we have the fimbria, and here we have again that seahorse structure, that C-shaped structure called hippocampus. So hippocampus is a seahorse structure. I'm going to show you why scientists and neuroscientists and neuroanatomists actually call it a seahorse structure. Of course, the reason is very clear, but I'm going to show you actually a comparison between hippocampus and a seahorse. It resembles a seahorse. This is the hippocampus. It's a convex elevation. We have convex, concave. It's a convex elevation in the middle portion of the anterior temporal lobe. So this is the temporal lobe. This is the anterior part of that temporal lobe, and hippocampus is located in the middle portion of the anterior temporal lobe. And it is approximately 5 centimeters long, and God knows how complicated this 5 centimeters long structure is in the brain. I mean, in the very last lecture, I'm going to talk about the macrocircuits of the hippocampus, and when I was studying that, I wondered how complicated the circuitry of the hippocampus is. Okay, so another way to talk about its position is that you see that cavity. I already told you that this cavity is the ventricular cavity. This is the inferior or temporal horn of the lateral ventricle. Okay, and the hippocampus is in this inferior or temporal horn of the lateral ventricle. And another thing about the hippocampus is that the hippocampus actually bulges laterally, so laterally means toward the side, or the sides. Medially means toward the middle part. So it bulges laterally, okay, in the temporal horn of the lateral ventricle. Sorry. Okay, this is a very beautiful picture. I told you that the hippocampus is a seahorse structure, and this is why it is called a seahorse structure. This is a real human hippocampus dissected out. You can see a part of the, the leg or the crus of the fornix, and also the fimbria here, and the hippocampus, the pes hippocampi here, the head of the hippocampus. And you can see that how it resembles the structure of the general shape of a seahorse. Very beautiful. Okay. The title, of course, I forgot to talk about the title. It's called hippocampal formation for a reason, because hippocampus is part of a bigger structure called hippocampal formation. And hippocampal formation has hippocampus proper, or the hippocampus itself, plus some associated cortical areas such as dentate gyrus, subicular cortex, parasubiculum, presubiculum, and subiculum, and also they're all interconnected with the entorhinal cortex. So this, and together they form a structure called hippocampal formation in the brain. Okay, fine. And let's talk about the fornix and the hippocampus again. So the hippocampus, has a head, of course, and the body and the tail. The head is slightly expanded, okay, relative to, the body and the tail, and it bears 2 or 3 grooves, okay, and these grooves are called pes hippocampi. Pes means paw or foot, okay, foot or paw, and those grooves in the head of the hippocampus, they resemble the paw of an animal, for example, a cat's paw. And I think in my model, let me show you with this fine dissector, you can see that we have 2 grooves here. We have 2 grooves that they resemble the paw of an animal. That's why they're called pes hippocampi. Okay. And then you can see that the hippocampus is interconnected with this C-shaped structure called fornix. And fornix is actually the main outflow bundle, the main nerve fibers that originate from the hippocampus and get out of the hippocampus. And this C-shaped structure is the fornix. Fornix is one of the commissural fibers of the brain. Commissural fiber is just a set of fibers, nerve fibers, that connect similar structures in 2 cerebral hemispheres together. And you can see that it starts from the tail of the hippocampus, and then it wraps around the thalamus. Okay, this part, its body wraps around the thalamus, and then we have the columns of the fornix that are connected to these mammillary bodies of the hypothalamus. Okay, and these 2 are this is amygdala, one of the structures related to hippocampus indirectly, and limbic system actually. These are the mammillary bodies of the hypothalamus. Okay, so this is the structure. So we have the columns of the fornix, then we have the body of the fornix. This part, this triangle here, is just a commissure of the fornix, connecting 2 fornix structures of the 2 hemispheres. And then we have the leg or the crus of the fornix, and then we have the fimbria. This is structure, this part, the fimbria of the fornix. I'm going to talk more about this structure. I've removed most of the labels, because these pictures are all labeled, just to practice neuroanatomy myself. So I'm going to talk more about it, but I just wanted to show you how the hippocampus bulges laterally. Okay, bulges laterally in the temporal lobe, in the temporal horn of the lateral ventricle. Yeah, I'm going to talk about different structures. For example, this is the optic nerve, the tail of the caudate nucleus, and this is the dentate gyrus here, and the hippocampus here, the subiculum, entorhinal cortex, and the fimbria here. And this is just the choroid plexus. Choroid plexus is a tissue is a type of tissue in the ventricles that is responsible for producing that CSF I told you about. Okay. This is another beautiful picture. You can see that those grooves in the head of the hippocampus and how they resemble the paw of an animal. That's why they're called pes hippocampi. And then we have this structure called fimbria. The alveus is the outer layer of the hippocampus. and the fibers of the alveus. I'm just going to tell you how hippocampus gives rise to, fornix. The outer layer of the hippocampus is called alveus. The fibers of the alveus, the nerve fibers of the alveus, they converge to form the fimbria. They converge together to form this fimbria here. And then, at a point beneath the splenium, so this is the corpus callosum, this is the splenium, this is the genu of the corpus callosum. At a point beneath the splenium of the corpus callosum, the fimbria and the fibers of the fimbria dissociate from the hippocampus in order to form the crus of the ipsilateral fornix. And by ipsilateral fornix, it means the fornix at the same side of the body, not at the opposite side. This is the crus of the fornix, and you can see that exactly beneath the splenium of the corpus callosum, the fibers of the fimbria are separated from the hippocampus in order to form that leg or crus of the ipsilateral fornix. And again, you can see the mammillary bodies of the hypothalamus. This is a part of the hypothalamus. This is the thalamus, and this is interthalamic adhesion. And this is the anterior part of the temporal lobe, and you can see again the location and the position of this hippocampus in the middle portion of the anterior temporal lobe. Okay, let's talk about limbic system. Well, hippocampus and hippocampal formation in general is part of the bigger system in the brain called the limbic system. Limbic system is very important for me because it was the introduction to the limbic system that got me into neuroscience. So, before the age of 17, I always wanted to become a cardiovascular surgeon. I didn't know why. I was just interested in the physiology and anatomy and surgery of the heart and the cardiovascular system. But there was a Korean drama on TV, and I was watching that, and there was a professor of neurosurgery in that drama, in that TV series. And in one episode, he was introducing the limbic system to high school students. And so he was talking about the roles of the limbic system in controlling our emotions, motivations, learnings, memories, and many other cognitive functions. And then he asked the student, those high school students, a very fundamental question that changed my life. He said that, okay, do you know where are the human's emotions? Or where they originate, where they come from, where our emotions are controlled? And the students said that, well, yes, from the human's heart. Well, it's a common belief that heart is associated with feelings, and that's why love, heart is a symbol of love, and when someone is sad, he or she says that my heart is broken. So it's a public belief that heart is associated with feelings. But we all know it's not. But the professor said, yeah, that's true. Our emotions come from our hearts. But do you know where is the human's heart? And that question was fantastic. He asked, do you know where is the human's heart? And the students, well, of course, they pointed to their chest. But the professor said, no. The real human's heart is here, is in the brain. All of our emotions, all of our feelings, all of our motivations, every single cognitive ability that makes us human beings, All of those are located here in the brain. So the human's brain is the real human's heart. The human's brain is the human, and human is the brain. And that changed me radically. And I was going to the kitchen, and I stopped and for a moment and listened to him and thought and told myself that he's right. All of our emotions, everything about that makes us who we are, is located here in the brain. And then after that, I decided that I'm going to be a neuroscientist. So that was a very short biography of me and how I got interested in the brain. So the limbic system is a very complicated system, like any other system in the brain, which is associated with the learning and memory, because the hippocampus and its associated structures are part of the limbic system. It's also part of the it also controls our emotions and feelings. For example, amygdala is part of the part of the limbic system, and it's associated with fear, the feeling of fear. And there are a lot of different structures. For example, we have the olfactory bulb and olfactory tract, and olfactory system is associated with the limbic system. We have the corpus callosum and the cingulate gyrus, and they are also associated with the limbic system. We have the thalamus, hypothalamus, and this is the hippocampus. This is a very beautiful structure called dentate gyrus. It has teeth-like edges, or tooth-like edges. That's why it is called dentate gyrus. The amygdala. So I just wanted to. I'm not going to talk a lot about the limbic system and all of these structures and connections between different structures here. I just wanted to tell you that hippocampus is part of a larger structure or system called the limbic system in the brain. Okay, so now let's get to that. This is that bulge. This is a picture showing how the hippocampus bulges laterally, and let's talk about some of the associated areas with the hippocampus. So this is a coronal section, and a coronal section or cot is just like this. I imagine that I have this, and I cut it like this. Okay, I cut it like this, and so I get the coronal section. And so this is a coronal section. We can see a lot of different structures here: the corpus callosum, the fornix, the choroid plexus of the lateral ventricles, the body of the caudate nucleus. Don't have the tail of the caudate nucleus. These are basal ganglia associated with movements. Also, scientists found that basal ganglia are associated with some cognitive functions as well. And this is the thalamus, and let's talk about these structures. So this is the optic nerve or optic tract, and this is the fimbria of the fornix and the hippocampus. This is the choroid plexus. I told you its function is to produce CSF in the lateral ventricle. And so this is the temporal horn of the lateral ventricle. And here we have those mammillary bodies. And finally, we have this entorhinal cortex, part of the medial temporal cortex, or temporal lobe, sorry. And we have subiculum, and you can see how the cortex bulges laterally. We have these structures called CA regions. These are zones of dense pyramidal cells, dense areas of dense zones of pyramidal cells in the hippocampus. I'm going to talk a lot about them actually later. And this is the dentate gyrus, another associated structure with the hippocampus. This is the dentate gyrus. Yeah, that's it. That's enough for this slide. Okay, now I want to talk about the disposition of cell fields. This is again a coronal section of the human's hippocampus, and this is stained by thionine. It's a dark dye for staining biological samples, and it's very beautiful because it shows the different layers of cortex. And so this is the entorhinal cortex. We have the subicular cortex here, and then we have that you can see the areas of the hippocampus. You can see these dense, very black areas. They're more black than the other areas, and these areas are, very dense in pyramidal cells. They have a lot of pyramidal cells and cell body of the pyramidal cells, of course. We have the dentate gyrus here and the fimbria. You can see how the outer layer, which is the alveus of the hippocampus, gives rise to fimbria. This is the same structure, but a schematic one. So we have parasubiculum, presubiculum, and subiculum, and then we have this different layers of the hippocampal cortex. So the outer layer is just the alveus. Then we have stratum oriens, stratum pyramidalis, stratum radiatum, stratum lacunosum moleculare, and moleculare. I'm not, I tried to find the correct pronunciation of these layers, but I couldn't find a reliable source. So I'm really sorry. I apologize if I'm pronouncing them not correctly. Okay, and this is the dentate gyrus. This is that. The reason it is called stratum pyramidalis is because the cell bodies of those pyramidal cells of the hippocampus are located in this layer. Okay, and these are different areas: the CA1 area, CA2 area, CA3 area, and CA4. They are called CA. CA stands for cornea-monas. Cornea-monas means literally means the horns of Ammon. Ammon was an ancient Egyptian god, and it had some round horns, exactly like this round structure of the hippocampus. That's why they're called cornea-monas. Okay, and we have different regions. The CA2 is the most has the most. It's very dense, actually. There's the most number of pyramidal cells. The pyramidal cells in CA3 are very large, the largest actually. So we have these layers, these areas and fields of cells. The reason I'm not explaining a lot about these areas is because I'm going to actually explain the synaptic circuits and the marker circuits of the hippocampus in the last lecture, so I'm just going to save it for them, for that lecture. And I just want to tell you a very general, give you a very general view of the hippocampus and its structures. Okay. Neuronal connections in the hippocampal formation. Well, we have different areas of the cortex. They all project to entorhinal cortex and subicular cortex. From, for example, cingulate gyrus, amygdala to the subiculum, and from different areas of the cortex to the entorhinal cortex. These pyramidal cells in the entorhinal cortex, they actually connect to directly to the subiculum and to these pyramidal cells of the CA1 area. They also connect to the granule cells of the dentate gyrus. Then these granule cells of the dentate gyrus, they connect to they make synaptic contacts with pyramidal cells in CA3 area via the Mossy fiber pathway. And then these CA3 areas, they send their axonal collaterals to CA1 pyramidal cells through the Schaffer collateral pathway. And then from the CA1 area, we have some outputs toward the fimbria. We have some, they contact the pyramidal cells in the entorhinal cortex and send information back to the entorhinal cortex and subiculum. This is a very general thing. I don't want to spoil it because I'm going to talk a lot about it in the last lecture. So this is just a very simple illustration of the neuronal connections in the hippocampus and hippocampal formation. We have the entorhinal cortex to different areas like CA1 and the dentate gyrus, and from the dentate gyrus we go to the CA3 area through the Mossy fiber pathway, and then from there via the Schaffer collateral pathway, pyramidal cells of the CA3 area contact the pyramidal cells in CA1, and then from there we go back to the entorhinal cortex or subiculum and the fimbria. Okay, so I told you that the entorhinal cortex, which is part of the medial temporal lobe, is a major source of projections to the hippocampus. And different areas, different cortical and subcortical areas, they project to this entorhinal cortex, and from there they project to the the sender inputs into the hippocampus. You can see that different parts of the cortical areas, they project to the entorhinal cortex. Their connections are maybe sometimes direct. For example, the insula directly contacts the entorhinal cortex. The superior temporal gyrus also do that. The cingulate gyrus, but some areas they are connected to the entorhinal cortex indirectly, and you can see different areas of various areas of cortex. They project to. They contact the entorhinal cortex, and these areas are actually numbered based on the Brodmann's classification. There was a neuroanatomist, and he numbered different areas of the cortex based on their function. Okay. This is a very beautiful drawing, again by, in one of his, one of the Dr. Frank's atlases. It's a processing circuit for recent memory. I'm just not going to tell you everything about this, but I'm just going to show you how different areas of the cortex and the hippocampus are interconnected with each other. So we have, for example, here we have the olfactory bulb, we have the amygdala, and they project to the entorhinal cortex, and then the entorhinal cortex connects to the hippocampal circuitry here, and the hippocampus sends its outputs toward the entorhinal cortex. And from the entorhinal cortex, those we have some projections to cortical areas like the primary visual cortex, primary auditory cortex, or somatosensory cortex. And this shows a very fundamental concept about memory. It tells us that memories, later I'm going to explain that in further detail, but memory is not stored in the hippocampus. Memory is formed in the hippocampus, and then it is transferred to different cortical areas to be stored. Okay, and there is. I'm going to tell you, talk about its, cellular mechanisms of how it is, how the memory is formed and then transferred to different cortical areas in the second lecture, second section of these lectures. Okay. Whew. So the hippocampal synaptic circuits. Well, I already explained that. I just wanted to show you that there are basically the pathway, the fibers from the entorhinal cortex to the different areas of the hippocampus They're called perforant. They form. They actually connect different areas, contact different areas of the hippocampus through the perforant pathway. And there is a direct pathway, as I told you, and there's a trisynaptic pathway. The direct pathway originates from layer 3 of the entorhinal cortex, and the pyramidal cells directly contact the very distal apical dendrites of pyramidal cells in CA1 area. Okay, you can see this connection. But there is a trisynaptic one, and the initial leg of this trisynaptic pathway starts and originates from the second layer of the entorhinal cortex, and the pyramidal cells in the second layer of the entorhinal cortex, they contact the granule cells in this dentate region or dentate gyrus, and then these granule cells contact the CA3 pyramidal cells through the Mossy fiber pathway. And then these CA3 pyramidal cells, they send their axonal collaterals to more proximal regions of these CA1 pyramidal cells through the Schaffer collateral pathway. Well, it is shown, as it is shown in this picture, the trisynaptic one is just a simple excitatory circuit. Later, I'm going to explain to you that it's not that simple, and it's not always excitatory. It depends on the state of activity in the hippocampus. Sometimes it can be the overall output of this trisynaptic pathway can be, inhibitory, and it depends on different things and different factors. I'm just going to explain that later. So. Yeah, this is a very general picture. And the reason it's called trisynaptic pathway is because we have three synapses. One from the pyramidal cells of the entorhinal cortex, layer 2 of the entorhinal cortex, to the granule cells. Then we have another one, from the Mossy fiber pathway, from granule cells to CA3 area. And then we have the Schaffer collateral pathway synapses. That's why it is called trisynaptic one. This is actually in a screenshot from a very beautiful and very accurate scientific animation about the structure of the hippocampus and hippocampal formation. I'm actually going to record a bonus video explaining that animation. And so again, you can see the coronal section of the hippocampus here, the subiculum, and you can see the CA1 area, CA2, CA3, and CA4. This is the dentate gyrus, and you can see how the outer layer of the hippocampus, the alveus, gives rise to fimbria, and it goes to, the fornix. This is the same synaptic pathway or circuit that I showed you. So we have from layer 3, we have a direct connection to CA1 from entorhinal cortex. From layer 2, we have connection to the subiculum, and then we go to the dentate gyrus. Sometimes subiculum directly contacts the CA3 area, but from the dentate gyrus, via the Mossy fiber, we connect to the CA4 area and CA3. There are some recurrent collaterals or recurrent or feedback loops or feedback connections in CA3 area. We don't have a lot of those feedback connections or recurrent connections in CA1 pyramidal cells, but we have a lot of those in those recurrent connections or feedback connections in CA3 pyramidal cells. I'm actually going to talk a lot about that in the last lecture, which is on the macrocircuits of the hippocampus. And then from the CA3, we go to CA1 through the Schaffer collateral pathway. And then the output of the subiculum and the CA1 area, CA3 and CA4, they all gather together in order to form the fimbria, and they go to the fornix. And sometimes the output of the CA1 area gets back to the entorhinal cortex, and from the entorhinal cortex it goes to other areas of the cortex. So this is again a very general but a very accurate diagram showing the hippocampal circuit. Okay, before talking about more about the synaptic circuitry in the hippocampus, and I'm going to talk about the LTP in the hippocampus and in different pathways of the hippocampus. I want to tell you, you know that better than me, but I want to still tell you about how the experiments are done on the circuits of the hippocampus. That was my question. I always had this question. Okay, how scientists actually find an area of the hippocampus and stimulate that and record from another area, and how they, work on that connectivity. So this is a living hippocampal slice. After killing the animal, the brain is actually is preserved in a very special medium that has all those things to keep the brain alive. And then we slice, and we get the hippocampal slice, and we put that hippocampal slice in a recording chamber. And we want to record from the pyramidal cells, so they need to stay alive. And so we actually put some special media in that recording chamber to keep the pyramidal cells alive, so that we can record from them. Okay? And so this is, for example, a stimulating electrode. I guess that this is the dentate gyrus. This is the CA4, CA3, CA1. So, yeah. Sorry. So this is a stimulating electrode. Sorry. That stimulates, for example, one pyramidal cell in one area of the hippocampus, and this is a recording electrode that records from another pyramidal cell in another area, and scientists actually try to figure out the connectivity between those areas and those different cells in different areas of the hippocampus. So this is a schematic view of that. Again, we have that hippocampal slice, and then scientists are stimulating the Schaffer collaterals in area CA3. And for example, what they do is that they record in the stratum pyramidal area, in CA1 area. So we have this is the stratum pyramidal. This layer is called stratum pyramidal because the cell body, or the pericardium, or the soma of those pyramidal cells are located here. And then we have stratum radiatum, the the region where the dendrites of those pyramidal cells are located. So scientists, what they do is that they stimulate a particular, for example, pyramidal cell in CA3 area, and they record from different layers of the CA1 area and different parts of the pyramidal cells in the CA1 area. I'm not going to talk a lot about these things here. These recordings here. So I just wanted to show you how scientists do that. The reason I showed you is because of this. I told myself that, okay, how they record this, how they do that, how they slice, and how they keep the neurons alive. So now I at least know the how, not exactly. I don't know how to do that myself, but at least I know how scientists do that. So this is the Schaffer collateral pathway, LTP. Before talking about this, let's talk about the LTP, as I didn't introduce LTP. Well, LTP, long-term potentiation, is it's all about those long-lasting changes in the strength of a synapse. So I already explained these. What do I mean? What scientists actually mean by the strength of a synapse? And it's all about how much the postsynaptic cell is influenced by or is activated or inhibited by the presynaptic terminal or presynaptic neuron. And both postsynaptic and presynaptic cells, they can contribute to the strength of the synapse. Sometimes in some synapses, the contribution of for example, postsynaptic neuron is much more than the contribution of presynaptic neuron. Sometimes the opposite happens. So now let's talk about the potentiation or the strengthening of a synapse in the Schaffer collateral pathway. Well, we know that the Schaffer collateral pathway is just a pathway from the CA3 pyramidal cells. They actually send their axonal collaterals to to proximal dendrites of CA1 pyramidal cells. And so actually what we do is that we apply a tetanic stimulation, and a tetanic stimulation is a very high frequency stimulation. For example, one tetanic stimulation lasts 1 second, and it has a frequency of 100 hertz. So it has 100 stimulations in 1 second. It's very high frequency stimulation. So we apply that tetanic stimulation and we record extracellularly. And we record EPSPs. When we record EPSPs extracellularly, those EPSPs are called field EPSPs. So now let's see what we have here. So this is EPSP, the level of the EPSP from the baseline. You can see that in the control, we want to know what is required, what is essential for the LTP in Schaffer collateral pathway in this synapse. In the control, when we don't do anything to these synapse, we have an LTP. First, here this arrow shows the time when we apply that tetanic stimulation. And here we have a very large LTP, very great LTP, and then it reduces because it's just one tetanic stimulation. It may last 2 or 3 hours, but then it is reduced to the basal level. But when we apply APV, APV is an antagonist for NMDA receptors. I'm going to explain, the structure of NMDA receptors and their mechanism and, how they're activated in the next lecture. But NMDA receptors are very important. They are ionotropic channels, and they are ligand gated, and their ligand is glutamate. So glutamate binds to the NMDA receptors, but they don't open until the cell is depolarized. Okay, I'm going to talk a lot about it. But APV, so APV is an antagonist. It means that it prevents the glutamate to bind to that NMDA receptor. So by doing that, it inhibits the NMDA receptors. It doesn't allow the NMDA receptors to be activated and to allow the ions to flow. So when we apply APV to this synapse, we have no long-term potentiation. So by this, we now know that NMDA receptors in CA1 area, in the pyramidal cells in CA1 area, are necessary for LTP, for induction and expression of LTP in this pathway, in Schaffer collateral pathway. Okay? Because when we inhibit NMDA receptors, we have no LTP. Now let's talk about the LTP in the direct perforant pathway. So the direct perforant pathway, we have the axons of the pyramidal cells of the entorhinal cortex, actually the third layer of the entorhinal cortex, directly which they directly contact the very distal apical dendrites of pyramidal cells in CA1. And what we do is that we apply APV, and we see that the long-term potentiation is not completely blocked. It's partially blocked. We still have a little bit of LTP. When we apply nitrendapine, And nitrendapine, APV inhibits NMDA receptors. Nitrendapine inhibits L-type voltage-gated calcium ion channels. When we apply both of them together, we have no LTP. So that means that in the direct perforant pathway, LTP depends partially on NMDA receptors and also on L-type voltage-gated calcium ion channels. Okay. So, and you know, in both of these pathways, this is the postsynaptic cell that contributes to the strengthening of the synapse. Let's see another scenario. The LTP in Mossy fiber pathway. So the Mossy fiber pathway is, starts with these granule cells in the dentate gyrus, and they contact the CA3 pyramidal cells. Let's see what happens. When we apply APV, nothing happens. But when we apply a protein kinase A inhibitor, which is in this case H89, it's a substance inhibiting the protein kinase A. In the presynaptic cell, we have no LTP. Let's see what happens when we actually do that. So when we apply a protein kinase A inhibitor, it inhibits the protein kinase A in the presynaptic cell. And that's why we don't have any LTP when we inhibit protein kinase A. Why do you think that, what's the role of protein kinase A? Well, when we apply that tanago stimulation, we increase the influx of calcium ion channels. The calcium ion channels activate calcium calmodulin. Calcium calmodulin activate the adenylyl cyclase, which is calcium calmodulin dependent adenylyl cyclase, and it produces CAMP. CAMP activates the protein kinase A by detaching the regulatory subunit and activating the catalytic subunit. And then the protein kinase A phosphorylates a synaptic vesicle protein called RIM1 alpha, which is associated with other proteins important for exocytosis and release of these synaptic vesicles. And so by doing that, protein kinase A actually augments the transmitter release. But when we inhibit that, we don't see any strengthening. We don't see the synapse to be strengthened a lot. So here you can see that the presynaptic cell has a major role in synaptic potentiation or strengthening the synapse. There are, of course, some NMDA receptors and AMPA receptors and other glutamate receptor in CA3 neurons, but their role in potentiation is very minor. Okay, so they're not very considered most of the time. So these protein kinase A's, they actually have a major role in inducing the LTP Mossy fiber pathway. Okay, so that was this lecture. I introduced the macroscopic structure and anatomy of the hippocampus. I talked maybe too much about the synaptic circuitry of the hippocampus, and at the end I explained the potentiation and what is necessary for long-term potentiation in different synaptic pathways of the hippocampus. In future lectures, in next lectures, I'm actually going to elaborate on these topics and subjects that I introduced in this lecture. So I hope you can watch those future lectures and other lectures as well. I hope you enjoyed this lecture. Well, that's it. Thank you so much for watching this video.